stem cells. The next category is really fun and interesting to talk about, and these are the stem cell products. So the stem cells are a subcategory of the HCTP, human tissue products, and these products are being marketed on the basis of having large numbers of stem cells from the donor that have a biological function leading to bone formation when implanted into a patient. Now, the processes by which these are procured and manufactured are all different from brand to brand. The claimed number of retained cells are different from brand to brand. But fundamentally, there are some real significant challenges with the whole concept here. One of which is, what cells are being delivered exactly? What is the function of those cells when they're implanted? What are they actually doing? And what is the demonstrated efficacy in terms of bone formation and, in our instance of what we're talking about here, spinal fusion? Because these are non-regulated HCTP products, there is no requirement for demonstration of safety and efficacy. Because these are human cells, they really can't be tested in normal animal models because of the histoincompatibility. The only way they could be tested in animal models is in athymic rats or other immunologically privileged or compromised animals, and as such, those are expensive models to do and are typically not done. So one of the claims that leads to the safety of this category of products is that mesenchymal stem cells, which are presumably the majority or some of the cells that are being transplanted, are immunologically privileged. What does that mean? An immunologically privileged cell is one that does not have surface antigens of the donor cadaver from which they came, and therefore should not be recognized or attacked by the immune system. In this rather interesting paper by Liu et al., they looked at the immunologic privilege of mesenchymal cells and found that they are, in fact, immunologically privileged. They will not be recognized as invaders when implanted. One of the challenges and problems with that, however, is that for these cells to function, and the idea, at least initially, was that they are going to differentiate down the lineage of forming osteoblasts, and they're going to, in fact, themselves participate in the bone formation process. Should these mesenchymal cells, in fact, differentiate down that or any other cell lineage, they will then express the surface antigens of their donor. They will then be recognized as foreign invader cells, and they will trigger an immune response. Most likely, they will be destroyed by T cells of the recipient, the recipient, and lead to potential complications associated with that immunological response. We don't really understand what is happening when these cells are implanted. We certainly don't understand what the efficacy is, and we don't understand what the safety is should these cells remain alive, as is claimed, and then remain alive inside of the recipient as well. While these stem cell products have now been marketed in the United States for well over a decade, there is surprisingly little published in the clinical literature to support their use. That in spite of the fact that on clinicaltrials.gov, there have been a number of clinical studies ongoing for most of that time. To date, there only have been 2 studies published, and both of those very recently. One of those was what appears to be a small cohort from a much larger study, 300 patients on clinicaltrials.gov, 33 patients in the publication. This was a prospective, non-randomized study that showed positive results, but only using plain film to evaluate fusion. This second study is a randomized trial conducted by people not sponsored by the manufacturer. This was an ACDF study and actually found inferior results in fusion with the stem cell product as compared to cancellous allograft. So to review the stem cell category, certainly the idea of taking a large number of stem cells from a donor, placing them into the patient to drive spinal fusion is a very compelling one. With the products that are currently commercially available, starting from recently deceased cadaver bone, removing the remaining viable cells, changing those or eliminating the cells that we don't want to end up with a subset of cells, cryogenically preserving them, thawing them and implanting them into the patient, really doesn't seem to be fulfilling that promise. So reminder, these are HCTP products, so there's no review for safety and efficacy from the FDA. To date, there is not compelling clinical evidence to support their use in the published literature, and if these cells are MSCs when implanted, and they should differentiate down the pathway towards becoming osteoblasts with the potential to form bone, they will express the surface antigens of their donor, and they will be destroyed by the patient's immune system, perhaps triggering some sort of a local immune response. So to date, spending almost the cost of BMP to use one of these products on the basis of the currently available evidence doesn't seem to be supported at this time.